Good afternoon, everyone. Welcome to the CBR Seminar series. I'm very happy to welcome Eric Cheng from my research group. I'm Karen Cheung. And for those of us who are on Zoom, I will be monitoring the chat in case anyone wants to pose questions. And for those of us in the lecture hall, if you could please turn on your microphone when posing a question, that would be very helpful so that people on Zoom can hear the question. And please remember to turn it off so that the next person can ask their question. Thank you.
Thank you. And thank you everyone for coming. I'm very excited to present my work on inkjet-based systems for single cell isolation for genomic analysis. There's a project we're calling the Isolatrix. So the Isolatrix is a benchtop cell handling instrument capable of partitioning cells into open well substrates. It uses inkjet nozzles to dispense droplets guided by a machine vision system using a neural network algorithm to accurately identify cells dispensing events. A unit of the instrument was installed at the Genome SAS Center, where it is being assessed to be integrated into their single cell sequencing pipeline. The instrument is compatible with most open well substrates, allowing for customized workflows, being an open platform capable of supporting many single cell applications, such as in monoclonal antibody development. However, the focus of the current work is on supporting single cell genomic sequencing applications. So why do we study single cells? Take cancer as an example. As tumor cells divide, they acquire genetic alterations, forming subpopulations with different genotypes and phenotypes. Evolutionary selective pressures, including the immune microenvironment and drug treatment, can differentially affect the survival of different subpopulations. Through single cell level sequencing, profiling of the genome, transcriptomes, and epigenomes of individual tumor cells can therefore reveal mechanisms underlying treatment resistance and metastatic progression, and pave the way towards improved treatments and clinical outcomes for patients. The first workflow which we will be looking to integrate our instrument into is the direct library preparation genomic sequencing protocol called DLP+, developed at BC Cancer. It uses a nanowell chip from Takara Bio, which is made of aluminum and contains an array of 72 by 72 nanowells. Each well is approximately 500 microns in diameter. Once single cell partitioning is done through dispensing the cell solution into the nanowell chip, the cells are lysed and select wells are tagged with barcodes for library construction, after which everything is pulled together to be sequenced. The genomic sequence of individual cells can then be reconstructed as each well has a unique barcode sequence. The use of open nanowell substrates allows for customized workflows to be developed, such as in full-length RNA sequencing or work in metabolomics, experiments which we aim to support in the future with our instrument. Looking at what is commercially available today for single cell systems reveals a trade-off in features. The 10x genomic system currently leads the market as it has high, although also highly variable throughput. It cannot verify single cell isolation and is a closed platform. Open emulsion-based competitor platforms exist, but suffer from variable capture efficiencies. Recent market competition from Cytina, Cell-N1, and the iCell8 provide improved cell isolation confidence through imaging, but cannot match 10x in throughput. Our innovation integrates high resolution imaging and neural network-based object recognition to enable high capture and high throughput single cell dispensing. The neural network assisted verification gives high confidence that genuine single cells are isolated, and rapid deposition into 5,000 nanowell open array enables customized workflows, enabling high throughput, high confidence, and cost-effective single cell omics. The core technology of our work is built around a piezoelectrically actuated inkjet nozzle. Inkjet nozzles are suitable for cell partitioning applications as they are drop on demand, capable of dispensing at high frequency in a highly repeatable process. Droplet volumes are tunable within a range of approximately 150 to 350 picoliters per droplet. And when coupled with high precision positioning instruments can achieve high spatial printing resolutions. The droplet ejection process is cell-friendly, demonstrating high dispensed cell viability due to low shear stress during the process. The glass capillaries used in inkjet channels allows for optical access to particles within the nozzle. Current inkjet cell printing systems uses a common approach where a camera will segment the nozzle into two regions. Typically, a region is identified near or around the nozzle orifice called the dispensing region. Wherever a cell is present in this region, it is identified to be dispensed in the next droplet. A second region adjacent to this dispensing region allows for the sedimentation of the cell into the dispensing region during machine movements. Therefore, the selection criteria for dispensing a single cell would be if the dispensing region contains a single cell and the sedimentation region is free of cells. Otherwise, the droplet will be discarded into a waste location. This approach, however, has drawbacks in that it tries to predict when a cell will be captured in the next droplet. It draws 2D regions into a 3D volume and requires calibration or prior knowledge to identify these two regions. And it only prints under ideal conditions, meaning that it can reject droplets that contain a single cell. Our approach, on the contrary, is a direct observation of the cell displacement within the nozzle after a droplet has been dispensed, where cell dispensing events are directly identified by comparing the images before and after a droplet. Therefore, no calibration steps are necessary, and every single cell dispensing event is captured. However, there are known drawbacks in inkjet cell printing systems. First, the cell encapsulation rates are governed by the Poisson distribution, where the events are skewed towards empty droplets that does not contain cells. The theoretical maximum for single cell rates is when lambda equals 1, which corresponds to a single cell rate of only 36%. However, lambda is a function of cell concentration and the droplet volume. And under normal operating cell concentrations, the majority of the droplets dispensed from any inkjet cell printing systems will be empty. Next, the common rate limiting step in cell printers is the machine movement time. As droplets can be ejected at high frequency, shown here in the chart is our instrument's relative times for dispensing droplets, image processing, and machine movement to an adjacent well. And you can see that the time required to dispense and analyze a droplet is significantly shorter than the time required for machine movements. Knowing this, our algorithm takes advantage of these two known limitations in order to achieve high throughput and accurate cell dispensing. The instrument positions the nozzle over the target well and continues to eject droplets until a cell dispensing event has occurred. This can be from dispensing a single cell or multiple cells. To meter the volume of droplets dispensed into each well, a timeout condition can be added where if a number of consecutive empty droplets are dispensed, the wells will be skipped and marked as containing no cells. Simulated results shown on the right of this print algorithm shows that single cell rates beyond the Poisson distribution can be achieved while minimizing machine movement time, allowing for high throughput. As the locations of the single cells are known within the chip, those wells can then be selected for further processing. Onto our instrument's hardware design. Here's the optical setup we use, where the imaging system contains of a camera and a zoom lens mounted in line with our nozzle, with the substrate moving below the printed area. The lens has a 14 times optical zoom used and has a working distance of 86 millimeters, allowing the printer to accommodate substrates with footprints of up to a standard well plate. The camera and lens has a pixel resolution of 0.697 micrometers per pixel and a maximum frame rate of 115 frames per second. As shown here in the video on the right, the imaging system is capable of resolving cells passing through our nozzle. Next, we have a 2-axis gantry system for positioning our substrate below the inkjet nozzle. It is stepper motor driven. It is a stepper motor driven linear stage with a microstep resolution of 0.496 micrometers and an accuracy of 50 microns. The high resolution stage allows for accurate positioning of the small features of the nanowell below the nozzle. This stage also has a top speed of 250 millimeters per second, allowing for fast traversal of the nanowell chip during printing. To secure the chip during printing, a custom holder was made using 3D printed parts. The top surface of the chip has to be kept unobstructed in order to make clearance for the nozzle moving above the substrate, and in addition to allow for the application of a microseal after printing. A FDM3 printed flexure spring made of PLA fully constrains the chip within the holder and secures the chip during printing operations. The single flexure spring also allows for access for installation and removal of the chip by the user. Our machine vision system is using a convolutional neural network that is used for predicting cell dispensing events. Temporal information is encoded into the input image of the nozzle. It incorporates past events into its considerations. The output of the neural network is a prediction score of 3 classes, with class 0 being a droplet that was dispensed that does not contain any cells, class 1 being a single cell was dispensed, and class 2 if multiple cells were dispensed in a droplet. The workflow for our instrument is laid out here. It starts with the user interface, where the user can access features to visualize the nozzle and droplets to assess their quality before printing. Next, an alignment step is run to align the nozzle with the nanowell chip. Then the print setup is used to define the area of the chip to be dispensed into, as well as other printing parameters, such as timeout conditions. Then the print routine is executed, and afterwards, the print results can be reviewed and select cells of interest are identified for sequencing. The alignment routine is laid out here, which is performed to align the nozzle with the small features of the nanowell chip, which is as small as 500 microns in diameter with a pitch of 542 microns. This routine uses water-sensitive paper placed near the substrate on the carriage. The routine works by first aligning the nozzle with the water-sensitive paper. Then the water-sensitive paper is aligned with a vertically mounted inspection camera we have on our instrument. Then 2 droplets dispensed 5 millimeters apart are dispensed on the water-sensitive paper, leaving a mark, which can then be identified by our inspection camera. As shown here, the water leaves a mark of blue and the camera identifies the droplet outline in green. Next, the position of the droplets, where the droplets are being dispensed into, is then used to align the 3 corners of the nanowell chip. And this is to account for any rotation in the chip, and it is compensated during the print run. After alignment, a print area is defined and it is highlighted by the red markers in this UI. The instrument will traverse this area in a raster pattern, and different print patterns can be defined other than a regular grid as shown on the left. Other patterns it can define is a no template control pattern, which it periodically skips a well. This can be used to assess any background ambient genomic signal that may be present by sequencing wells that do not have anything dispensed into them. In addition, a checkerboard pattern can be defined to assess any cross-contamination from the printing process itself by sequencing neighboring wells that should not contain any of the dispensed droplets. The bottom images shown here shows a replication of the print patterns on water-sensitive paper, which turns blue when exposed to liquid. The convolutional neural network deployed on our instrument was trained using TensorFlow and optimized through hyperparameter tuning with Keras Tuner. As shown in the confusion matrix here, there perhaps is unsurprisingly underperformance in the class 2. This is due to underrepresentation in the training data set due to the Poisson distribution. Therefore, a major challenge for us is finding a way to rapidly generate reliably labeled data to train our neural network on. This has led to the development of our microscope-based labeling pipeline for the rapid generation of accurate labels for training our neural net. It begins with printing a grid of single droplets on a microscope slide. The cell sample is stained with CSFE, which remains fluorescent even though the cell has dried. The cell suspension medium, which in this case is PBS, dries and leaves a salt residue feature, allowing us to identify where the droplets have been dispensed on the slide. The slide is imaged under fluorescent and bright field microscopy with our Nikon TI2E microscope. And imaging of the printed grid allows for correlation of the microscopy images to our instruments captured images. Using this approach, due to the cell encapsulation process being governed by the Poisson distribution, the most commonly occurring class of no cells being in a droplet can be rapidly identified through a lack of fluorescent signal. Droplets of interest containing fluorescent signal can then be investigated manually and used to determine the correct class. So the first step in this pipeline is a reconstruction of the printed area on the microscope slide through stitching of images taken under 4x magnification under both bright field and fluorescent channels. By stitching and performing background subtraction, we can better localize the droplets on the printed slides, allowing for droplet segmentation. The stitch 4x image shown here is composed of two parts: the printed grid, which contains an array of 50 by 20 single droplets. And on the bottom right is a barcode. This barcode is used to keep track of the microscope slides and for referencing with the captured printer images. The barcode zoomed in here is a 6 by 6 grid used to encode in binary the date and time in which the slide was printed. 6 bits of data is required to accommodate the minutes unit. The topper of the barcode is a timing pattern of 101010 for the reading algorithm to know the spacing of the grid. Then below it is a year, month, day, hour, and minute information. So for example, this barcode and this slide was printed on August 25th of this year at 3:11 p.m. From the 4x stitch image, we also have fluorescent readings, which should correlate to where the cells are dispensed on our slide. So the pixel locations of these fluorescent signals are then identified and due to its location in the image, is used to correlate to a physical location on our microscope. And this allows us to selectively image droplets of interest, so droplets which should contain cells under higher magnification. We use Christopher's algorithm to traverse the cell-containing droplet grid, which is an approximation of the optimal path to the traveling salesman problem, to move the microscope under fluorescent and bright field imaging to capture higher magnification images under 20x magnification. So once we have all this information, it is aggregated into our labeling user interface. You can see here at the top is the 4x stitched image. The droplets of interest to be labeled are highlighted in the gray box. So these are droplets that have been imaged under 20x magnification. The current display droplet is highlighted by the red box. The image is also interactive, so that by clicking on the highlighted regions, a droplet can be reviewed, labeled, or relabeled. To the right is the 20x magnification images of bright field and fluorescent channels overlaid together, and as well as the isolated fluorescent channels. This image is used to verify the presence and count of the cells within a droplet. Below is the corresponding image from our printer, which is to be labeled. This workflow allows for rapid automatic labeling of class 0 events, the most common occurring events, in addition to a data-rich user interface to generate accurate labels for training our neural network. In the future, this setup can be expanded to assist in the labeling of other events such as cellular debris from primary samples. Another approach we are taking to increase our class 2 representation is to generate realistic looking training images using generative adversarial networks, where two competing fully convolutional networks compete against each other. So the generator takes a noise vector input and tries to generate realistic looking images, while the discriminator is given a task of determining if it was a real or generated image. Our GAN is trained using both class 1 and class 2 data, and this work was done in PyTorch. Here we have some sample generated images after training, along with the generator and discriminator scores. As the generator improves, it is succeeding in fooling the discriminator, and when the discriminator score falls below 0.5, the generator is successfully fooling the discriminator, as the discriminator's accuracy is no better than a coin flip guess. Taking a closer look at some of the generated images, and by splitting the RGB channels from these color images, we can see that the GAN does a good job in replicating the displacement of cells within our droplet, which has a droplet being ejected between each frame. In fact, the image series on the top right is a real class 2 image. So in the future, we'll be looking to include these GAN generated images into our training data set to improve the representation of the underrepresented class 2. So now onto printing experiments done into nanowells. This following work was carried out at the Genome Sciences Center. For this experiment, a selected area of 20 complete rows were printed starting at row 28. The time of condition was set to 5 droplets. The cell sample used here was the 184H mammary epithelial cell line, a test cell line used at the Genome Sciences Center. You can see from the bottom 2 images, the cell suspension in a cell counter, and a plot of the cell size distribution. The cell size distribution was quite large, which is a good test of how well our neural network can generalize between different cell types. And here we have a playback of the print run. And during a print run, the user interface will print in a raster pattern and update the grid display in real time. And shown here is being played back at 8 times speed. And as the markers are being populated, the color represents the cell printing events. Blue being no cells, so reaching the time of condition, green representing a single cell, and yellow representing multiple cells. The marker color darkens as the number of droplets dispensed into each well increases. In total, the print run of 140 wells took 260 seconds, which is 4 minutes and 20 seconds, with an average time of 0.181 seconds per well. In total, we have counted 881 single cells dispensed in this experiment. After a print run, the cell dispensing events can be reviewed by clicking on a well of interest. When a well is selected, the UI will display the corresponding neural network prediction scores along with the corresponding image pairs from our nozzle. Here in the yellow box, I display a microscopy result of a cell calling image routine, which is a quality control step as part of the Genome Sciences Center single cell sequencing pipeline. As the nanowell chip is made of aluminum, the chip is opaque for imaging and has to be placed upside down over an inverted microscope to capture epifluorescence signal of the stained cells. So as a result, the column order in this image here is displayed here is reversed. So here we have a well that has been identified as being empty, having reached the time of condition. The neural network prediction score indicates this, along with the corresponding image pairs, as well as the microscope images. The well next to it contains a single cell, again shown in the microscopic images and our neural network prediction scores, as well as the next adjacent well at the edge of the chip. So there is good agreement between the cell calling images that is obtained through microscopy, an automated microscopy routine, and the neural network prediction scores and our nozzle images. Here, the cells from the previous slide are shown and manually re-imaged to better show the cells in the well. The bottom image, the green channel shows the H-church cells, and the grayscale color channel is showing the concave bottom of the nanowells. This was captured through reflected light of the excitation light source without an emission filter. So here are the neural network predictions and nozzle images of other select wells. On the left, there's an instance of class 0 events where a number of cells within the field, where there are a number of cells within the field of view. Next to it is a class 1 event of a large cell. And on the right are 2 examples of class 2 events of cell doublets. With the additional dimensional data from our neural network, the user also has the ability through the UI to perform metric-based filtering of the printed single cells, such as applying a threshold to the prediction score to select cells for further library construction. So here now I'll talk about some of the ongoing work for our printer development, which includes an environment controller, including a temperature-controlled substrate. To do this, we have a Peltier thermoelectric element, the hot side of which is regulated by a custom 3D printed liquid cooling channel. Liquid cooling is favorable in this case, as opposed to the more common active heatsink, as we do not want a fan blowing near our sample. This unit is capable of maintaining set point stability within a plus or minus 0.5 degrees Celsius range over 30 minutes, accommodating enough time for a complete print run. And as shown in the plots given here, there are 2 common set point temperatures displayed of 4 degrees Celsius and 37 degrees. In addition to regulate the substrate temperature, the ability to control the relative humidity within the instrument is also of interest. For example, in maintaining dew point to prevent evaporation of the materials dispensed, such as the cell solution, or perhaps lysis buffer, which may be preloaded into the chips. However, humidification with small droplets are desirable in order to prevent the deposition of external genomic material onto the nanowell sample. Therefore, we use dry fog humidification using a modified airbrush. Dry fog is defined as droplets of less than 10 microns in diameter and has been demonstrated to reduce condensation and surface wetting. Relative humidity of over 97% using our spray head has been achieved in a test enclosure. To verify that our droplets are in the dry fog regime, an experiment spraying atomized water onto water-sensitive paper was performed. Due to the small size of the features, reflected light microscopy was used to image the exposed paper. After accommodating for droplet spread factor, an average droplet diameter from our spray head was measured to be 7.85 microns. Lastly, we are working on a method of extracting the dispensed cell features, such as cell size and circularity. This can be used to supplement bioinformatic analysis. This begins with the segmentation of cells from the nozzle images. To achieve this, we take advantage of the cell feature finding capabilities of our trained neural network. A modified image was passed through just the convolutional layers of our CNN. The resulting feature map is then collapsed into a binary image shown here in black and white. And it does a good job in effectively localizing cells within our images, as highlighted in the red circles. These features can then be used as masks to segment the cells to be further processed so that features can be extracted from them. This is leading towards a UI element to be developed where the users can visualize and select cells of interest for analysis, shown here in the functional mockup on the right. And so, yeah, so our future work, we are working towards improving our neural network classification accuracy through our microscopy labeling pipeline, as well as our GAN. And we are looking to perform full-scale genomic sequencing analysis at our partner site at the Genome Science Center, beginning with Agilent Analyzer QC to assess the fragmented DNA quality. Future work, we hope to support full-length RNA sequencing, as well as other omics experiments, and as well as to support the identification through our neural network of cellular debris within the cell sample. Thank you very much. So yeah, any questions? Yeah.
Yeah, Eric, it's really great to see all this together in one presentation. I've seen bits and pieces of it over the years. So I think one sort of major question is, if you a lot of people would say, well, why don't you just use flow cytometry? It can do single cell dispensing. And I was wondering if you use flow cytometry for this purpose, what kind of problems would you run into?
So I believe the question was why do we not use flow cytometry for single cell analysis? So this is not my area of expertise, but as I understand, flow cytometry has drawbacks in that it cannot be isolated into these nanowell features, which really takes advantage of some of the small volume chemistries used in this analysis. And as well as I believe there's a lack of quality control measures in flow cytometry. So feedback that we hear a lot is that there's really a lacking of assurance of single cells being isolated, which is why we took a machine vision approach capable of logging and extracting as much data as possible of the cell sample that was isolated.
And is your goal then ultimately to not do is your goal to ultimately not do the verification? You have the verification from the side, but then to confirm that you look at the nanowell substrate. So is the goal to then not emit nanowells at the end once it's sufficiently high quality?
So the ultimate goal is if in the imaging step of the nanowell after printing, it's is eliminated through the assurance of our neural network. This we did as part of the workflow that is established at the Genome Science Center, which is a genomic sequencing workflows. But for some experiments, this imaging step is not possible as, for example, in sensitive experiments like RNA sequencing, the chip might be preloaded with lysis buffer. So in which case, if there is multiple cells dispensed into a well, that, as far as I know, would not be able to be differentiated at this imaging step and until later for the genomic analysis that might have some clues, but there's uncertainty there as well.
Thank you. We have a question from Professor Lansworth. Peter?
Yeah, hi, Eric. A nice talk. So this is Peter Lanzer, BC Cancer Research Center. I guess in your current setup, the concentration of the cells in your dispenser is very important, right? If you have too diluted suspension, you have too many empty wells. And if you have too much, it might be very hard to find wells which have just a single cell. So I was wondering, is it possible to come up with a situation where using your networks, you identify when the single cell is present and only then allow that cell to make it to the well, because otherwise you're going to end up with these wells which have no cells or too many cells. Could you just have some kind of, I guess, valve which would basically only allow single cells to go through, or what are your thoughts on that possibility?
Yeah, that's a very interesting question. Our instrument was not designed to do this kind of single cell selection, in that all the droplets are sort of captured or dispensed into the chip. However, there is work that we're hoping to do in the future of tuning finding out what the optimal cell concentration would be in order to maximize the single cell rates. But yeah, that's a very interesting question. I think if there's a specific use case in mind, it'll be very interesting to talk afterwards. Yeah.
Thanks.
What's your predicted occupancy for single cell?
So we have simulated results, and so these don't take into account of sedimentation or other factors that may affect the cell dispensing, but I believe up to 85% was simulated, up to 85% single cell occupancy.
What's the smallest cell size you can sort, or droplet size? How small can you go?
Yeah, that's a great question. So we've recently started working with a PBMC cell sample, which our cell counter told us has an average size of 6 microns. So quite on the smaller side of cells. We have designed our optical system so it has a resolution of about 0.6 microns. When we first designed the system, we have also in mind to work with cell nuclei, but we have not tested that out yet.
Have you tried bacteria at all?
No, we have not. I think bacteria can has quite a large range of possible cell size, even beyond or below a micron, I believe. Or so that we have not tried that.
A micron would be pretty close, but it'd be very interesting. I mean, for a lot of the environmental area size of things, and there's a lot of interest in doing those kind of things. So that would be another application for your instrument if you can get down there.
Yeah, that's a very interesting. We have not tried it. A micron might be close to our detection limit, but it'll be interesting to try. Yeah.
That was a really good talk, and your whole lab should be complimented and congratulated on some wonderful technology being developed. And I'm just going to follow up on the last two questions, which are really starting now to touch upon the biology and the applications here. And you really did start to think about this in one of your lines on future directions, fragmentation of DNA. And how are you going to make sure about the quality of the cells that you're putting in here? Is this technology messing up the quality of these cells? How are you going to make sure about this? What's going on?
Yeah, that's a great question. And it definitely is not my area of expertise, which is why we have partnered with researchers at the Genome Science Center, including bioinformaticians, technicians, chemists. So a very thorough analysis of the readout data from our instrument is somewhere in the near future. Yeah.
So that was PBMCs in that little movie that you showed?
H-terts.
I'm sorry?
184 H-terts. It's a
I see. Because there was a fair amount of heterogeneity in those cells, and even it looked like there was some blebbing going on there.
Yeah, so the from our cell counter, there was a very large range in the cell size in that sample. And our goal is to have as general of a neural network for different cell sizes or cell types as possible.
Thanks, Ed. When you mentioned messing the quality of the cells, in what way do you mean? What are your metrics? What are your metrics for the quality of the cell?
Well, if there are issues going on with the cells changing during the course of this analysis, we wouldn't get the same answers as one would expect. So if the cell is changing as a consequence of this analysis, we would be having all DNA was fragmenting, methylation was occurring, other events might be. So we would have to
Oh yes, of course. And I think which is our reasoning for making the instrument as high throughput as possible. So as the cells are not exposed to an unfavorable environment. So for example, the instrument currently used in the Genome Science Center single cell workflow, I think a common benchmark they quote is 1,000 cells in 45 minutes to an hour. So we can do that in a significant small amount of time in probably less than 10 minutes. So that on our end, our mitigation strategy is to try to go as fast as possible, at least for now. And then later on, we will definitely be assessing different factors that might affect sequencing quality. Yeah.
Any other questions? I don't see any questions in the chat, so thank you everyone for coming.
Thank you.